I'll go ahead and get started. Hopefully, people fill in. Hello, everyone. Welcome to this session of ARCS Forward in the National Scholar Speaker Series, cleverly titled He Cells, She Cells, and T Cells, How Sex Affects Immune Response. Today, we'll be focused on immunology and specifically about immune responses in the lungs and how biological sex affects them. Thank you for joining us. I am Kevin Yehl, and I'll be moderating our session today. So, I'm a scholar alum, having received my ARCS award from the Atlanta chapter about 10 years ago in 2014. Crazy to think about that. Time flies, I guess, when you're having fun. But currently, I'm a professor at Miami University, Miami in Ohio, and I am a professor in the biochemistry and chemistry department. My research focuses on developing phage therapies for combating antibiotic resistance. And my lab also develops biosensors and point-of-care diagnostics using nucleic acid technology. So it is my pleasure to moderate this session and reconnect with the ARCS Foundation as being an ARC award winner. So ARC stands for Achievement Rewards for College Scientists, and ARCS Forward is the National Scholar Speaker Series, which features young scientists from across the ARCS Foundation network of 15 chapters. Wow. And today we have two excellent scholars doing research in the area of immunology. And so the mission of the ARCS Foundation is to advance science in the United States by providing financial awards to academically outstanding U.S. citizens studying to complete degrees in STEM. And so the ARCS Foundation has a 65-year history of furthering this mission and is a leader in scientific philanthropy. So on behalf of the ARCS Forward Committee, thank you for joining us today. So now to the stars of the show. Let me introduce our two esteemed guest speakers from two opposite sides of the United States. Today we'll hear from Cameron Mattingly, a scholar from the ARCS Atlanta. And then we'll also hear from Caroline Duncombe, a scholar from ARCS Seattle. So welcome both of you, and thank you for taking time to speak with us today. So first, let's hear from Cameron. Take it away, Cameron.
Okay. Can everybody see my screen okay, Kevin?
Yes, it is working, and we can hear you perfectly well. I will mute
Perfect. Thank you. So hi, everyone. I'm Cameron Mattingly. I'm an immunology graduate student at Emory University in Jake Kohlmeyer's lab. And I'm excited to talk to you all today about human lung tissue-resident CD8 memory T cells and how they protect against respiratory viruses. So of the title, I am the T cell portion of the He cells, She cells, T cells. So first, just a quick outline of my talk today. So I'm going to start by going over just some basics of immunology. So the immune system is so complex and very fascinating. And so I want to give you a little overview of, what immunology is. And then I'll get a little bit into my research on CD8 T cells in the human lung and how they protect against respiratory viruses. So immunology is the study of the immune system, and which is basically a whole network of cells and proteins that sort of act like an army to protect you from harm. And this whole network is split into two main branches, as you can sort of see in the chart shown here. So those branches are the innate immune system and the adaptive immune system. And so innate immunity is the first line of defense against pathogens. It's called the innate system because it's a defense that all animals naturally have. And some parts have been around for more than 500 million years. And the innate system is made up of physical and chemical barriers like your skin or enzymes in your stomach. But if a pathogen gets past those defenses, then the innate system is also made up of specific cells and proteins. Also, the innate system is able to recognize a lot of different pathogens in a very broad, non-specific manner. So it doesn't know that what exactly you're infected with, but it does know that an invader is present. And there's lots of cells that make up the innate immunity branch, but the main function of these cells is to patrol tissue sites throughout the body, start fighting off the infection, and then call for backup from the adaptive immune system if it needs it. And so adaptive immunity has lots of key features, but one of its main features is its ability to recognize and respond to specific pathogen. So it's able to say, oh yeah, that virus there, that's swine flu, or that bacteria, that's tuberculosis. So they're able to recognize specific features of pathogens because the cells are extremely diverse. Basically, for each pathogen in the world that could possibly exist, your body needs to have at least one cell that can recognize and respond to it. So in this way, the cells are able to adapt to whatever enemy is thrown your way, and it's thus a very effective strategy since they're so specifically trained. And so when we think of the adaptive immune system, in the broadest sense, there are two main cell types, B cells and T cells. And there's also different types of T cells, but today we'll be focusing in on CD8 T cells. So what role do B and T cells play in the immune response? So B cells, you may have heard of, because they mainly produce antibodies. And so these antibodies are able to bind to viruses and prevent infection. CD8 T cells, on the other hand, are also known as killer T cells. Instead of binding to the outside of the virus, basically they're able to recognize infected cells. So once they recognize an infected cell, the CD8 T cell can bind to it and directly kill that cell so it prevents the infection from spreading. Additionally, CD8 T cells can also produce cytokines or messenger proteins that can help warn nearby cells of the infection. So the combination of these adaptive immune cells allows you to prevent more cells from getting infected and clears infected cells. Ultimately, those two together help you get better faster. So the adaptive immune system is really great at clearing infections. So why have the innate immune system at all? So the adapted cells are very slow to respond. So T and B cells aren't necessarily at the site of infection. And so basically they can reside in lymph nodes. And so say you stepped on a nail covered in bacteria. That bacteria first has to be recognized by innate immune system at that site of infection. Then innate cells have to travel to the nearest lymph node. And at that point, they can then activate T and B cells in the lymph node. And then the T and B cells take time to form their army and then travel back to the site of infection. So ultimately, the innate cells are just a lot faster at responding due to their location and are necessary to hold off the infection for a while until the adaptive response can get there. So what does this look like in real time? So say you breathe in a respiratory virus and it's able to infect epithelial cells in your lungs. So your innate immune system is going to spring into action within minutes to hours and start the immune response. If it fails to completely clear the virus, it's going to call for backup by taking a sample of that virus and transporting it to the lymph nodes so it can activate B and T cells. Then it can take, so this process here, this travel process can take hours again. And then T and B cells will ready their armies and travel back to the site of infection. So you're not really, so these adaptive immune cells are not really arriving at the site of infection until 4 to 7 days post-infection. But then once they're there, they're able to eliminate the pathogen quickly. But this whole process is why it takes time to get better, even when you get just something as simple as a common cold virus. So now that we've covered some basics of immunology, we can talk more about my research on the role that T cells play in the immune response to respiratory viruses. So despite the availability of a seasonal vaccine, influenza and other respiratory viruses still remain a great burden to human health. So over the past 18 flu seasons, the vaccine has been on average around 40% effective. So it's still helpful to get, but there's definitely room for improvement. And that's because the current flu vaccine primarily produces antibodies. And so while these antibodies are very helpful in lots of ways, there's also a drawback to them. So the antibodies primarily bind surface proteins on the outside of the flu virus. But these surface proteins are what frequently change year to year. And when this happens, the antibodies can no longer bind to the flu virus and the vaccine isn't very effective. So the effectiveness of the current flu vaccines basically relies on experts perfectly predicting which strains will circulate that year. So really a lot could be improved for flu shots, but ideally we'd want a shot that generates an immune response that is more durable and can fight against different strains of the flu. And so that's where CD8 T cells come in. So CD8 T cells and B cells work differently. So while antibodies bind to the outside of the flu virus, CD8 T cells bind to or recognize internal flu virus proteins. So the good part about this is that these internal proteins are more conserved across the different flu strains. So even though you get changes in the outside of the flu virus, the internal proteins stay the same. And so the protection CD8 T cells provide isn't as dependent on predicting the right strain of flu each season. And CD8s could even protect against strains of flu that aren't even in existence yet. However, our current flu shots don't generate CD8 T cells, so we're missing out on this added protection. Another key part of the adaptive immune system is their ability to form what we call memory. So this is true for B and T cells. So in this graph, the black line here is the infection, and the gray line here is the CD8 T cell response. So along the x-axis here, we have time after infection, and along the y-axis is the cell or pathogen density, depending on whether you're looking at the black or gray line. So you can see when we get infected with the virus for the first time, it takes a little bit of time for this gray line, the CD8 T cells, to respond. So they need to be activated and grow in numbers. But around 1 to 2 weeks, you have a large amount of CD8 T cells that can fight against that virus, and the amount of virus starts to decline in response to this. And then once the virus starts to decline, we don't need as many CD8 T cells around, and so their numbers start to die off too. This decrease in T cells is important because the immune response requires a lot of energy. Think how tired you feel when you get sick. So once the enemy is gone, all the troops are no longer needed. However, a small subset of these CD8 T cells remain behind as memory CD8 T cells. And these memory CD8 T cells are already trained and ready to go. So if your body gets infected with the same virus again, in this yellow panel here, you can see that you don't have the same lag period to generate that CD8 response. So the CD8 T cells can basically more quickly clear the infection. And there are different types of memory CD8 T cells, and we can categorize them based off where they circulate throughout the body. But my research focuses on tissue-resident memory CD8 T cells, which don't recirculate through the body and instead remain localized within certain tissues. And so these tissue-resident memory T cells, which we commonly abbreviate as TRM, they form at common sites of pathogen entry, such as lung, skin, gut, and genital tract. And since these TRM don't recirculate throughout the body, they can act as sentinels of the immune system. This means that since TRM are in the tissue at the site of infection, they don't have to take time to travel, to activate, and thus have a rapid response time. And based on what we know about CD8 lung TRM, they're really useful against fighting against respiratory viruses and are worth exploring more since CD8 TRM could be particularly useful in a vaccine scenario. However, most of what we know in research is from animal models. So one of the challenges in studying TRM is that since they're not in the blood, it's not easy to sample them in humans. So overall, the research on these cells is technically challenging. So here is a schematic of a TRM cell at work in the lung. I just want to orient you a little bit. So on the left here are airway epithelial cells. And then on the right is a blood vessel, and in the middle are TRM and other immune cells in your lung tissue. So one of the main ways CD8 TRM help in an infection is communicating with other cells by producing messenger proteins or cytokines. So that's all these little blue dots and arrows and lots of abbreviations here. But basically they serve to recruit immune cells to the site of infection, so that could be from the blood, and then also activate local cells that are already in the lung tissue so that they can begin readying their defenses. And one of these main antiviral proteins that CD8 TRM produce is interferon gamma. So interferon gamma is important because it acts as an alarm system, warning nearby cells of danger. So when a cell receives this interferon gamma signal, cells ramp up their defenses by producing various proteins that make it much harder for viruses to enter and replicate within them. And again, while we know a lot from animal models like mice, we know very little of how they function in the human lung. So I decided to look at how does interferon gamma from human lung TRM impact local cells, and does it reduce the amount of virus in an influenza infection? So the first step in this research is obtaining human lung cells. So this can be a major obstacle for a lot of people, but our lab collaborates with LifeLink of Georgia. So this organization works to procure organs for transplant. However, not all organs are able to be transplanted. Sometimes there are areas of acute damage from physical trauma, or sometimes a match can't be found. So our lab receives these other healthy lungs that are unable to be transplanted, and then we can process these lungs, and this provides us with a rich repository of lung cells that we can then use for experiments to investigate the function of CD8-TRM. And so the first step in my work is understanding the type of CD8 memory T cells each of my donors have. So again, CD8 T cells only respond to specific pathogens. So we're looking at whether each donor has any CD8 T cell that could respond to a flu infection. So here we take flu peptides or fragments of flu proteins, mix them with lung cells, and if the CD8 T cells produce interferon gamma, then we know that they would respond to a flu infection. So when we do this for all 22 lung donors, we observe a range of CD8 T cell responses. So here in this graph, each donor is a specific symbol and color. Along the x-axis, we see all the different viruses we looked at, and along the y-axis is the frequency of CD8 T cells that could produce interferon gamma. So basically, the symbol is above this very tiny dashed line. That donor has a memory CD8 T cell that could respond to that specific virus. And we can see that all these donors here in this red box have CD8 T cells that could respond to a flu infection. So once I confirm that our lungs had CD8 T cells that could respond to flu, I can move on to looking at how those cells impacted the cells around them in the immune response. So for this, I again mix my lung cells with fragments of the flu virus. This activates the CD8 T cell. The CD8 T cell can then produce interferon gamma, and then the interferon gamma can act on other local lung cells. So I identified different types of immune cells and epithelial cells in the lung, and then measured their activation profile by basically looking at certain proteins that these cells produce. Then, to determine whether the activation I see is due to interferon gamma, I add in a reagent that blocks interferon gamma. So looking at the difference in activation profile with and without interferon gamma blocked tells us whether interferon gamma is responsible for the activation we see. So one of these activation proteins I'm looking at is called ICAM1. Basically, this molecule is involved in cell adhesion, which is important for immune cells interacting with one another. And here I'm looking at the expression of ICAM1 on epithelial cells. And so the histograms, these sort of gray plots here, show the amount of epithelial cells that have this ICAM1 protein. And it also shows it for the 3 different treatment conditions that we looked at. So when the CD8 T cells are not activated, there's a baseline level of ICAM1. When we activate our CD8 T cells with the flu proteins, we get an increase in ICAM1 expression on the epithelial cells. And then when we block interferon gamma, so this anti-interferon gamma, here we see that ICAM1 levels remain similar to baseline levels. So basically what this tells us is that in a flu infection, the interferon gamma from CD8 T cells activates epithelial cells in the lungs. And so we decided to then look at these epithelial cells more. And so there's lots of different types of epithelial cells in the lung that play an important role in the function and the immune defenses. So we have ciliated cells with these hair-like projections. We have secretory cells like goblet cells in green here, which produce mucus. And we also have basal cells, which can grow into secretory or ciliated cells. And so what we were interested in is if all epithelial cells were affected to the same degree by interferon gamma. And so up until this point, we use surface proteins on epithelial cells to look at activation. But what we really wanted to get at was whether or not there's something happening at the gene level to help mediate protection. So when a cell produces certain genes, that becomes proteins, and it can help protect them against the virus. And so basically, we, again, activated our CD8 T cells and then separated out epithelial cells. And when we do this, we can look at genes that are involved in the antiviral response. So all these genes in the bottom here are involved in the defense against a virus. And then we're looking at them by the different cell type here. So basically, the more red the circle, the greater that gene is expressed in that cell type. And so what we see is that when we look at these antiviral genes, when the CD8s are activated with flu, basal cells have the greatest change in these genes, while we really don't see much activation of the ciliated cells. And so up until this point, we see that human lungs CD8 TRM can produce interferon gamma, and this interferon gamma activates epithelial cells. But does this activation alter the susceptibility to influenza infection? So to correctly answer this question, we needed to infect lung epithelial cells with the flu. So for this, we set up a culture system of epithelial cells, similar to how they grow in the lungs. So in this model, we grow epithelial cells so that they're in contact with air on one side and media on the other side that represents the lung tissue. And when we grow these epithelial cells this way, you get all the different types of epithelial cells, and you also get mucus production like you would in the lungs. And once we had all our epithelial cell culture, I then infected them with an H1N1 strain of influenza. And then we looked at whether interferon gamma could basically reduce susceptibility to infection. So here we had different treatment groups, so with and without infection and with and without interferon gamma. Then 18 hours following infection, we collected all these cells to perform different experiments to look at the activation and infection levels. And we first looked at the expression of ICAM1 on epithelial cells. We see that compared to uninfected, and when we have infection, you don't really get an increase in ICAM1 protein on epithelial cells. However, when you treat with interferon gamma in this blue and green group, you get a significant increase in ICAM1 expression. And it's possible that this tells us is that 18 hours post-infection, interferon gamma can activate epithelial cells and possibly expedite the immune response to get faster viral clearance. We also wanted to look at what interferon gamma does to reduce the amount of infection. So here I'm looking at the frequency of infected epithelial cells on the y-axis, and we see that compared to epithelial cells that were infected with no interferon gamma treatment in yellow, if we treat with interferon gamma in blue and green here, we see that they have significantly less infection, showing us that interferon gamma from CD8 T cells does help protect against influenza infection. And so just to sort of summarize, in a flu infection, these memory CD8 T cells rapidly respond and produce interferon gamma. Interferon gamma is then able to activate the lung epithelial cells and helps prepare epithelial cells, especially basal epithelial cells, to prepare for a viral infection. And this interferon gamma activation of epithelial cells reduces the susceptibility of influenza infection. So as a result of this research, we have a better understanding of tissue-resident cells in our lungs and their function in the immune response to influenza. And this information can then be used to help better design vaccines and prevent the spread of respiratory viruses. So with that, I just want to thank all the members of the Kohlmeyer Lab for their help along the way with this research. I also want to give a special thank to organ donors and their families. And lastly, I really want to thank all of ARCS and ARCS Atlanta for supporting me on my educational journey. Not only has the financial support helped ease the burden of graduate school, but knowing that there are people in the community that support young scientists really helps me, keep driving with my research. And it's really helps to know that they're, cheering for me to succeed. So I hope you learned a little bit about the immune system today, and I'd be happy to take questions later. Thank you.
Wow, thank you very much, Cameron. What a tour de force. So now let's hear from Caroline. I'll let you introduce yourself.
Hi, everyone. I just need to share my screen, I believe. Hello, everyone. I am Caroline Duncombe. I'm a PhD candidate here at the University of Washington in Seattle, and I study the impact of biological sex on vaccine responses, specifically to a malaria vaccine. First, I want to introduce you to the concept of how biological sex influences immunity. Biological sex can be divided into kind of like three biological like metrics. So there are sex hormones, which have different levels of expression of X and Y linked genes that can alter your immune response. Additionally, there are sex hormones, which are which I will be focusing most of this talk on, but they are small molecules that can really also influence your immune response. And then there are also environmental and microbiota differences between males and females that can also influence your immune responses. Overall, these three factors ultimately result in general, this is a generalization, there are always exceptions in biology, that females tend to respond better than males to almost all existing vaccines by at least one metric in which we measure the efficacy or immunogenicity of different vaccine responses. And so there are lots of reasons for that, and will be delving really into my research, which is how sex hormones modulate this response and make males respond worse to vaccines. So let's go back a little bit and talk about what sex hormones actually are. I'm sure everyone has their own kind of relationship to what do they define sex hormones as, but I looked it up in the dictionary, and according to the Merriam-Webster dictionary, a sex hormone is produced especially by the ovaries, testes, or adrenal cortex and affects the growth or function of the reproductive organs or the development of a secondary sex characteristic. And while this is technically a right definition, I kind of disagree, or not disagree. I would like to redefine and add some definitions to what I would say a sex hormone is from like this biological researcher perspective, which is that sex hormones are produced especially by those organs, but not only by those organs. Sex hormones are actually produced within other tissues as well. They are small molecules. They're produced in many other organs. They are involved in a whole bunch of functions beyond that of reproductive capacity. They differ significantly between people, but everyone has all sex hormones. They just exist at different levels. And so let's go into a little bit more of the specifics. So this is the molecule testosterone. So when I'm talking about a small molecule, I'm talking about this kind of like moiety that kind of scares a lot of people in organic chemistry. And it's kind of, but this is what testosterone looks like. And testosterone exists within this pathway of hormones where every sex hormone is derived from cholesterol. So cholesterol has this kind of rate limiting step where it goes and converts into a progesterone, and then progesterone is converted into an androgen, and then androgens, which is where testosterone is located, is then converted into estrogen. And so when I say that your body is able to produce all of these hormones in many different organs, this enzymatic process happens within many different parts of your body. And so while I know this is an immunology course and I'm an immunologist, I also think it's really interesting to think about the intersection of endocrinology with immunology. And so here are all the different organs in which you might actually see more of an influence of hormone biosynthesis. And later on in this talk, I'll be focusing actually a lot on the liver. And the liver is a really important endocrine organ and is responsible for a lot of different components. But you can see that there are a lot of different organs within your body that influence your hormone levels. And so how do these hormones actually function? Well, this is a schematic that's showing that the sex hormones or steroids can work via actually like 4 different mechanisms. And I'm not going to get too much into the detail here, but there are mechanisms in which hormones directly bind and alter the transcription of your, like the function of all cells, but it also is able to act within these kind of like, we call them non-genomic pathways that can also alter the responses of your cells. And this also happens within T cells. So there are 4 main facts that I want you guys to all take away from this talk about sex hormones, because it's a subject I'm very passionate about. And I think there's a lot of misconceptions about how sex hormones might view what they are. And the first is that sex hormones exist within these complex pathways, and they enact over like thousands of different functions within your body, and they modulate a bunch of different areas, including your immune responses. Almost every cell in your body is dynamically regulated by sex hormones. And as I mentioned before, every person has every sex hormone, regardless of biological sex, and it's the levels that differ. And non-reproductive tissues also play a crucial role in the synthesis of sex hormones. My research really focuses on how every cell in your body is dynamically regulated by sex hormones, and really focusing on how this alters the immune cell function, and specifically T cell function, and how the liver is the location of hormone metabolism will be another focus. So ultimately, sex hormones modulate the immune cell function, and I'll just be focusing mostly on how these hormones alter CD8 T cell function. Okay, so now we'll go into, after that background, about a little bit about how biological sex and sex hormones can influence your immune response. I'll be talking about how sex hormones are altering the immune response to the vaccine I work with, which is a malaria vaccine. I work within a vaccinology lab. My goal of my PI is to create an efficacious malaria vaccine that is effective in all populations. And so to give a little background about what malaria is malaria is a mosquito-borne infection that's transmitted by mosquitoes. Africa accounts for about 95% of all cases of malaria disease. There are a lot of accelerating factors to malaria, and actually cases of malaria, like mortality and disease, has been increasing ever since the COVID-19 pandemic started. And there are also other factors like climate change and drug resistance that are affecting this kind of actually reversal of all of these amazing treatments that or advancements that malaria researchers and advocates have been trying to achieve in reducing burden, but has really not been able to. And so while these numbers are kind of old from 2020, I will note that they've been increasing recently. And so malaria, or Plasmodium, which is the parasite that causes malaria disease, undergoes this complex life cycle where I'm sure everyone knows that it starts with the bite of a female mosquito, and female mosquitoes inject these Plasmodium parasites, which I'm going to introduce to the term of sporozoites. Sporozoites is the form, or the initial form, of Plasmodium, and these sporozoites are really smart. They can enter into your skin, and then they know to enter your bloodstream, and they kind of like migrate via this like tactile motility, and they home to your liver. And then once they enter the liver, they live there for about 6 days within humans. And then I use mouse models, so it takes about 2 days within mouse models. And they home into your liver and they affect your liver cells or hepatocytes. And during this process, they actually start to replicate a lot. And then after 2 days or 6 days, they enter into the bloodstream where actual clinical symptoms of malaria begin. And so there's kind of this asymptomatic phase during the liver, which I think many people don't realize is happening. And so that's why when you get infected with malaria, you actually start getting symptoms of it about a week later. Because then during this blood stage process, they really start to replicate. And then eventually, if a mosquito happens to bite you again, they will take up that parasite in its sexually mature form. And that's how onward transmission occurs. And so my lab focuses on creating a liver stage malaria vaccine that actually clears all these parasites while they're in the liver stage prior to blood stage infection. This is actually a really great way to address I like to think about them as the parasites are kind of like sitting ducks, because in the liver, they're kind of just replicating and they're not causing any disease. And so they're in the same spot, and they're really much more easy to target during this phase than when they're in the bloodstream and there's so many more of these parasites. And it also blocks the progression of clinical symptoms and en route transmission. And so how do you clear your liver cells that are infected with Plasmodium? Well, luckily, there's been decades of research that have identified that actually a really great mechanism to clear these infected hepatocytes is by inducing memory CD8 T cells, which luckily, because Cameron did such a great job of explaining. I don't have to re-explain what a memory CD8 T cell is, and actually also specifically tissue-resident memory CD8 T cells, which are these CD8 T cells that are able to quickly home and kill these infected hepatocytes. So here you can see this is in the liver, kind of, we call it liver sinusoid, but it's kind of like your blood vessels that are running throughout your liver. And they kind of migrate and they look and look and look for an infected hepatocyte. And they're able to like quickly release all of these kind of defense mechanisms or cytokines or perforin, and they're able to kill that infected hepatocyte. I'm actually going to show you, this is a real image that I take. This is the technique called microscopy, where I'm able to stain plasmodium, or the replicating parasite. And so this is actually an infected hepatocyte. And you can see that these T cells, this is after 44 hours of incubation within a mouse liver, that these T cells have identified this infected hepatocyte and are attacking it. And you can see this rupturing kind of this cell. And so this is a real-time image of these T cells attacking and clearing an infected hepatocyte. Okay, so one way in which our lab has developed a vaccine strategy that can induce kind of these CD8 T cells to be able to quickly target and clear the infected hepatocytes is by using what we call a prime and trap strategy. And so we use mouse models to test our vaccines. And so I'm just going to take you through a little bit of how this mouse strategy works. So it starts with priming memory CD8 T cells. And we do this priming step with a device called a gene gun that is a DNA type of vaccine. And so gene guns are really interesting. They have these gold particles that are coated with DNA. And we attach this kind of gun type moiety, though it's not, we call it a gun, but it's really just hooked up to a helium tank. And we shoot a burst of helium, so it creates like a high level of pressure, and it injects all of these really small gold particles into the skin. And then your skin immune cells are able to pick up that DNA and start to develop a memory response to what we're trying to have it target, which is actually a protein that coats the surface of Plasmodium parasites. And so this is the priming step. The second step is, because we want to induce these kind of more tissue resident memory CD8 T cells in the liver, we trap these CD8 T cells by delivering and leveraging the unique ability of these sporozoites to home to the liver. And so what happens here is we actually radiation attenuate, basically disrupting all the DNA to make sure that the parasite is unable to replicate. And after this kind of attenuation process, we are able to like use the ability of the sporozoite and to migrate into the liver, but then is not able to go on. But it is able to kind of like bring together and really target the liver to induce these kind of memory CD8 T cell populations. And then this is a model that we're using to test a vaccine. And so you have to have kind of, how do you test it? And here, what you'll see in my data is that there's kind of this challenge step, which is where we're testing whether the vaccine worked or didn't work. And we're challenging by giving the mice virulent Plasmodium parasites, or here it's Plasmodium yewellii, which is a rodent form of Plasmodium that I'm using within this model. And so we're challenging them with these virulent parasites to see if the vaccine worked or didn't work. And so the question that I started with in my PhD was, what is the effect of biological sex on this vaccine strategy? Well, in our lab, and this is kind of a well-known fact in liver stage malaria field, is that almost all preclinical vaccine work is done in female mice. There are many reasons for that, but probably one of them is that you can protect them. You see here, this you'll start seeing a lot of these graphs throughout the rest of my presentation, where this is percent of protected mice. So 0 means that none of the mice were protected. So all of them ended up getting a blood stage infection, which is our metric for non-protection. And if you see this dark, it means that they were protected. So 100% of the female mice that were vaccinated with prime and trap strategy were protected from infection. And then here you can see within our naive group, so these mice were not vaccinated, that all of them, so our like infectivity control, all of them ended up having infection. So this works within female mice. But within male mice, when you do the exact same vaccine strategy, you actually see this reduction where now you're only protecting about 15% of male mice from malaria blood stage infection. And you can see here that on this imaging, so this is kind of attaching kind of like a fluorescent GFP or GF, sorry, it's not GFP, it's luciferase. And you can see like relative burden within these mice. And the males are, while they have reduced infection compared to these unvaccinated mice, they aren't able to clear infection. And it only takes one infected hepatocyte or one infected liver cell to cause blood stage infection, which means that the vaccine did not work. And so how do sex hormones alter this outcome? Well, one way to alter sex hormones within mouse models is by performing surgery on them and removing actually the ovaries or the testes. And so here what we're doing is an ovarectomy, which is removing the source of estrogen and progesterone within these mice prior to vaccination. And so when we performed this kind of reduction of ovarian female hormones, you can see within these female mice that there was no difference in protection between, so 8 out of the 10 mice were protected still, and so we're seeing high levels of protection. But then also within the mice that received a sham surgery, which is an equivalent level of surgery, but without actual removal of the ovaries, that there's not a significant difference in protection outcome between those. But this is actually, this model, if one of like the known effects of estrogen is that it increases protection outcomes. And so if you were to look at kind of like a suboptimal vaccine strategy by removing this initial priming step, and so you can now see that there's actually a benefit of protection. So what I want to point out here is that while the ovarian hormones aren't altering they aren't decreasing protection outcomes, they're actually increasing your protection. So there's actually a beneficial effect of estrogen and progesterone to vaccination. And so now the next question is, what is the effect of androgens or testosterone? Well, testosterone can be reduced by performing a castration, or here we call it an orchiectomy. And so by performing a castration, you can see now that in these male mice that normally from a sham surgery, they have very low levels of protection. When you remove their source of testosterone, you are completely reversing this protection outcome. So you're seeing that testosterone is actually a major modulator of protection within these mice. And so I work within a vaccinology lab. And we tried to increase and try to overcome this barrier and leverage the effect. And we found that by increasing this kind of like priming step from the vaccine, we saw no difference in protection. And then when we tried to increase the responses to the trapping step, once again, we saw no difference in protection. You can just see in these graphs that there was not a significant increase in protection outcomes. We're really looking for that like dark bar here that indicates protection. And also I did all of this complex immunology work on characterizing like the quality and the quantity of these memory CD8 T cells. And once again, I found no difference. And so my thesis is really trying to figure out like, why aren't we able to protect these male mice? Like, what is happening here? Well, I did a simple experiment where instead of performing the surgery or the castration prior to vaccination, I performed it simply before challenge. And so you can see from this data that by performing the surgery simply before a challenge, once again, you're seeing that complete reversal of protection. And so ultimately, testosterone levels at time of challenge produce protection to prime and trap vaccination. And this really indicates that what's really happening here is maybe not the fact that, biological sex is influencing these memory CD8 T cells during vaccination, but it's really influencing these CD8 T cells during its kind of effector phase, when it's trying to quickly home to these infected hepatocytes and clear them. And so I like to think about this model where it's like biological sex can influence many different functions, and there are direct and indirect effects. And this is really all happening during this effector phase. And so in conclusion, sex hormones are dynamic modulators of your immune response. Males and female mice respond differently to prime and trap vaccination. And it is testosterone at time of challenge that reduces protection from virulent sporozoite challenge. And ultimately, this connects back to the point that overall, it seems to be that sex hormones are major modulators of your immune response. And ultimately, this influences the fact that females respond better to almost all existing vaccines on average. I will say on average. And so thank you. I would like to thank many people within my lab. Dr. Sean Murphy and Melanie Shears are my mentors and PIs here at the University of Washington, have really been a core part of supporting this research effort, and the University of Washington. And I also want to thank the ARCS Fellowship. It's been really a great support to have funding to pursue my interests. And one of the really great parts that I've been able to pursue, I think in part because of the freedom to not have to worry too much about finances, is I am actually also, I've been able to really pursue my interest in science communication. And very recently, if you're ever in Seattle, I produce actually a science-themed improv show where we invite scientists to give TED Talks on their research, and then have our team of 10 professional improvisers pull and create improv off of that talk. And this has been going on for over 2 years now and has met a lot of success in Seattle. And I really think ARCS has been an important role in supporting me in this endeavor. So thank you.
Wow, thank you, Caroline. And that was fantastic. And thank you, Cameron, as well. So that was great. The immune system is so fascinating, yet very complicated. So it was great to have those insights. So let me see if we have a few questions in the chat, and if not, I have plenty of questions. I always have questions. So I guess we'll start off with I guess tell us, how did you become interested in science and at what age? And I guess we could do it in reverse order, or whoever wants to go first.
Yeah, I can go first. I actually kind of think I'm different from a lot of people in that I didn't know I was going to be a scientist until I accidentally took a chemistry course in college and realized I loved it and was good at it, and loved trying to figure out what was the fundamental mechanisms behind how the world works. So it really was something that I unexpectedly stumbled upon, in part because of that chemistry class. Yeah, it's really amazing how single professors can really shape how and what you end up becoming.
Yay, chemistry. So what about you, Cameron?
Yeah, so I always liked science just growing up in general. And so I did biology in undergrad. But it wasn't until I did joined a research lab and got into research that I really knew that that's what I wanted to do. And just to sort of agree with what Caroline was saying, having just one really impactful mentor can really shape how you see the progression of your life and everything. And so that happened definitely at during undergrad, and then continued to happen with all my mentors since then.
Great to hear. So I guess I'll start with you, Cameron. I have a couple sciencey questions. So, do any current vaccines generate CD8 T cells? And I'll add in, where do RNA vaccines fit in this immune response?
So I mentioned that, current flu vaccines mostly just generate the B cells and the antibodies. They technically generate some CD4 T cells that I didn't talk about today, but they don't generate any CD8 T cells. And that's mostly because of the type of vaccine it is. It's like a protein vaccine, but it's also because of the route of vaccination. So if we were to give so the current flu vaccines are given intramuscularly. So, you go and get a shot in your arm. But there are types of intranasal vaccines that could generate these CD8 TRM. And so research on that is definitely progressing to try to help make them better. As far as like the RNA vaccines, so the current SARS-CoV-2 shots do, as far as I'm aware, generate some CD8 T cells, not necessarily the tissue resident cells that you would have in your respiratory tract, though. And that is because it is an RNA vaccine, basically just like the mechanism of action or the function. When you get the shot, the RNA is able to trigger a CD8 T cell response, and then you have those memory cells that live on afterwards.
Okay. And then, do our CD8 T cells memory ever get too full? Like, do we ever run out of memory?
Full, yeah. Yeah, so there is a concept that you only have the space for so many CD8 T cells or so many of a certain type of cell in your body at certain locations. It's a resource thing, and that's why. So they do decline like after an infection because you don't need all of them there all the time because that will just like take too much energy to maintain. And they do T cells can decline over time as well. So there is some evidence that there could be a population limit, but any research that's been conducted so far, we've not seen a limit at all, So
Thanks. And then, I guess one last question I have is, what if you put someone else's CD8 T cells that's like, I guess, responsive to the flu in another person? Like,
Yeah, so there's some so I will start with things that wouldn't work. There's some, things I guess I would need to explain. So basically, just like you have a blood type, there's also something called an HLA type. And so your HLA type is something like they match in transplant. So you have to match the HLA type to get an organ transplanted in. And the HLA type is really a set of certain immune proteins. And so if your HLA type doesn't match another person's HLA type, you'll generate an immune response against that. And so you'll be generating this huge immune response, and it would be worse for you because you just basically your body would be attacking those T cells that got put into you instead of them being helpful for a future response to like influenza or something like that. So ultimately that's not probably a path to go down, but that does work with antibody transfers. So the antibody transfers don't have those certain proteins, and that's why you can get antibodies transferred between people.
okay, well, thanks. So no T cells in people. Ooh. So I guess, Caroline, I have a couple questions for you. So why is it a female mosquito that causes the disease? Is it hormone related?
Yeah, so it's not hormone related. Well, maybe mosquito hormone related. So female mosquitoes need to like give birth to more mosquitoes. And so they actually pick up some really important compounds from humans to help them like create eggs to be able to like I don't like give birth is probably not the right word for it, but to propagate their progeny. And so that's actually why mosquitoes are the ones who will be biting you. Males don't bite, male mosquitoes don't bite. Humans.
I didn't know that. Interesting.
Yeah, it's only female mosquito. So whenever you're like hitting that mosquito, it is most likely a female.
You're hitting a female mosquito.
Yeah,
Okay.
you are. Yeah, I like to sometimes make the joke that like male mosquitoes are like moral support for female mosquitoes who are the leading like animal killer in the world.
Wow.
So there's kind of a whole part of it.
That's pretty funny. I guess maybe I learned that fact a long time in biology, but I forgot.
Yeah.
Interesting. And then so for the mouse observations, does that translate to humans for like I guess are males more susceptible to malaria?
Yeah, so actually, males have higher prevalence of blood stage infection than females do. So, males in general are unable to clear like the infection once it starts, or it takes them longer. It takes them about twice as long to clear a blood stage infection. And so, yeah, this does kind of translate into humans in terms of malaria burden. And then to the vaccine response, honestly, one of the great and stressful things about studying the impact of biological sex on vaccine responses is that many clinical trials right now for these types of vaccines aren't powered to account for like capturing that sex difference. And so it's sometimes really hard to tell because of poor data quality in humans.
Okay, thanks. And then this leads me to my last question is, are there drugs to reduce testosterone, or is this like an active drug discovery pursuit?
Yeah, so there are drugs to reduce testosterone. I will be honest, I don't think that will be a very popular treatment strategy to increase vaccine responses. As much as I would love to point to the science behind it, realistically, I don't think that's going to happen. There are drugs to reduce testosterone for other biological diseases and for other reasons. People actually use a compound that is used for male contraceptive. So if you decrease testosterone levels, it also decreases your like sperm counts and stuff like that. Not sperm counts, like the ability to be fertile.
Yeah.
And so yeah, so that's kind of
Okay.
yeah.
Well, yeah, well, thank you both. I think we're wrapping up on time. I have a couple closing remarks. I first like to thank you both, Cameron and Caroline, for speaking with us today. That was a lot of fun. Always great to learn new science. I learned a lot. The immune system is critical in phage therapy, and this is something I definitely need to learn more about. So your research is fascinating and very important to answering many scientific questions surrounding response to disease. And so the ARCS Atlanta and the ARCS Seattle chapters are very proud of your research. And so it's these kind of cutting edge scholars that each of the 15 chapters of ARCS Foundation supports. ARCS awards make a huge impact on these scientists, as we heard from both Caroline and Cameron. And believe me, I know my ARCS award came at a time towards the end of my PhD studies where I was in a dire need of a new computer. My hard drive was dying. I was like, if I lose all this data, I don't know how I'll be able to write up my dissertation. Also, just the prestige of the award helped me get a postdoc at MIT, which was an awesome experience. And then that helped me land my dream job here as an assistant professor. So thank you so much to ARCS for your support. And then lastly, the ARCS gave me the confidence I needed. So I don't know if there's any graduate students on this call, but I know that the process can be very challenging, but there is light at the end of the tunnel, and your guys and girls are doing great work. So thank you everyone for tuning into this session. We'll send out a link to the recording for you to share with others. Please share. You'll also receive a short survey, so please let us know if there is a topic of interest to you. We really do use these surveys to design our sessions, so please return them and complete them. And then please plan to tune in on September 24th. Mark your calendars, September 24th at noon for a special Hall of Fame session with our newest inductee, biostatistician, Dr. Paula Deer. So remember, mark your calendars, and thank you very much. And thank you for tuning in, and have a wonderful summer.